presenting to us today. Dr. Luo focuses on understanding and engineering viral mechanisms to develop new therapies for cardiac disease, and as well as neurologic disorders. And she's also engaged in developing oncolytic viruses for cancer therapy, which I didn't know about, and we're doing work on that, so I'm hoping we can collaborate. Her presentation is on mastering immune chaos, strategic innovations for viral myocarditis via the innate pathway modulation. And I think we've all received the learning objectives, which are very clearly stated. Thank you, Honglin. And we look forward to your presentation, and then we'll have some time afterwards to engage with you and ask some questions. So look forward to that. So Honglin, if you'd like to grab control of the screen again, brilliant. And
okay, so can you see my screen?
I can see it really clearly. I'm going to mute myself.
Okay, that's great. So you can hear me clearly too. Okay, so yeah, thank you. Yeah, thank you, David. Yeah, today I'd like to give an update about our research on viral myocarditis. So in this talk, I'm going to focus on our recent research in terms of the innate immune mechanism of viral myocarditis. So I'm going to divide my talk into 3 main sections. Oops. So first, I'm going to give some background introduction about viral myocarditis, including the most recent, the COVID-19 related myocarditis. Then I'm going to talk about briefly about our research on the mechanism of virus-induced direct damage to the myocardium. So I will mostly focus on the virus-encoded protease. What's the contribution to the direct damage to the myocardium? So the major part of my talk will be focused on the innate immune mechanism of viral myocarditis. So I'm going to introduce our recent work on inflammasomes, including two inflammasome. We focused on NLRP3 and CAR8, as well as our ongoing research, cGAS and STIM pathway. So myocarditis is defined as a non-ischemic inflammatory disease of the heart. So in addition to various infections agents, including viruses, bacteria, fungi, parasites, and many non-infectious agents have been identified, including toxins, and also systematic inflammation, such as the many autoimmune disease has been linked to myocarditis, vaccinations, such as the smallpox vaccine or influenza vaccine. And also recently, the COVID vaccine has all been linked to, one of the side effects is linked to myocarditis. I also like to point out here, one of the most severe side effects using immune checkpoint inhibitor is myocarditis. The incidence is low, but the death rate is quite high. It's up to 50%. So viral infections are the most common cause of myocarditis. According to the 2019 Global Burden of Disease Project, so the global prevalence of viral myocarditis is about 10 to 106 patients per 100K individuals. So we know viral myocarditis affects all the age groups and both female and males, but it's more common in young adults, and also it's more common in male versus female. So likely due to the male always have more severe inflammation as compared to female. And clinically, viral myocarditis can resolve on its own without any treatment, but up to 30% of patients diagnosed with myocarditis can end up developing dilated cardiomyopathy and heart failure. So many viral causes have been identified over past 70 years. So the prominent one, including the internal viruses and the adenoviruses, the parvovirus, herpes virus, and the most recently the coronavirus, including SARS-CoV and SARS-CoV-2. Among all of these viruses, I like to point out internal virus as the based or extensive studied virus, because that's the first virus identified associated with viral myocarditis, and also because of the availability of the animal models. So many of the virus, it's only clinical evidence suggests that may play a role in viral myocarditis, but there is a lack of animal models. So diagnosis of viral myocarditis is a combination of clinical symptoms and also various clinical examinations, and sometimes endomyocardial biopsy. So among different clinical techniques, cardiac MRI is considered these days as the non-invasive gold standard for the diagnosis of myocarditis. So, but for the mRNA, which can provide, can detect the inflammation, fibrosis, and provide information about the cardiac function. So, but for a definitive diagnosis of myocarditis, the heart biopsy is needed. So it's Dallas criteria that's considered as the gold standard. So the EMB provide evidence of non-ischemic myocyte necrosis, as well as inflammatory infiltration by both H&E staining and also immunostaining using different immune cell markers. So in addition, the biopsy can also allow us to identify the viral causes. So by PCR or in situ hybridization that allow us to detect the viral genome, and also by immunohistochemistry staining that allow us to detect the viral protein. So since the outbreak of COVID-19, there is a significant increased reported COVID-19 related myocarditis. So the incidence, I just searched this morning about the incidence COVID-19 related myocarditis. It's about 150 to 4,000 cases per 100,000 individuals. So current understanding of the mechanism of COVID-19 related myocarditis is mainly based on the autopsy or the cardiac mRNA findings, because there is a lack of animal models. So available data suggests that the COVID-19 related myocarditis is related to myocardial inflation and also the systemic cytokine storm. So H&E staining and the IHC staining shows clearly the infiltration of inflammatory cells. So many macrophage, less T cells. And also there is still an ongoing debate with regard to whether SARS-CoV-2 can directly infect or cause damage to the myocardium. So some in vitro data using the human iPSC-derived cardiomyocytes shows when the virus titer is high enough, they shows the cardiomyocytes can be directly infected and causing damage and decrease contractility of the cardiomyocytes and the inflammation. However, most of the autopsy samples shows no strong evidence the virus can directly target the cardiomyocytes. And also finally, the common pathological changes observed in post-mortem COVID-19 heart is the microthrombi, which is believed to contribute to cardiomyocyte necrosis. So the myocarditis is considered as a triphasic disease. The phase 1 is occurring between 0 to day 5, is acute viremic stage. So this stage is characterized by prominent viral replication and virus-induced direct damage of the heart. And phase 2 is a subacute inflammatory stage, is characterized by the infiltration of immune cells, early stage macrophage or NK cells, later T cells, B cells, and profound production of inflammatory cytokines, including interleukin-1, 6, and TNF-alpha. And also this stage, we can observe the clearance, the virus start to be cleared from the heart, and as well as immune mediated myocardial injury. So phase 3 is the chronic remodeling phase. We can observe the fibrosis, stenotic cardiomyopathy. Usually this stage, the virus already cleared. So usually we cannot detect viral particle or very low viral persistence. So this stage is right now current is quite established that both virus-induced direct damage and also immune-mediated damage both contribute to the pathogenesis and development of viral myocarditis. So despite of the much efforts and also many clinical trials, there is still no clinically proven treatment for acute myocarditis. Current treatment still mainly based on the supportive treatment, improve the cardiac function or using the anti-arrhythmia drug. So in terms of the, we know for a long time virus can induce direct damage of the heart. So Coxsackie virus B3 is an internal virus. So Coxsackie virus B3 induced myocarditis mouse model. Coxsackie virus B3 has been widely used as a model system to study viral myocarditis, both in vivo and in vitro. So about 30 years ago, Dr. Bruce McManus has reported that CVB3 infection of the immune deficient mice can cause severe myocarditis without an effective immune response. So his early study suggested that the virus can directly damage the myocardium. So this slide shows the sequential slide sections for both in situ hybridization and H&E staining. As you can see, the viral genome positive cardiomyocytes also got injured, suggests the direct damage occur after viral infection. So accumulated studies have suggested virus-encoded protease is an important contributing factor to virus-induced direct damage to the myocardium. So internal viruses encode two proteases, 2A and 3C. The primary function of these two proteins is to process the polyprotein into individual structure and non-structure protein, which is a critical step during viral life cycle. And we also people also found these two proteases can cleave the many host proteins for viral benefits to enhance viral application. So early study by Dr. Kirk-Lawton published in Nature Medicine in 1999. So he first reported that 2A can directly cleave dystrophin. Dystrophin is the muscle cytoskeleton protein important to maintain the muscle integrity. So mutation of dystrophin has been linked to Duchenne muscular dystrophy. So he reported 2A viral protease can directly cleave the dystrophin in the heart. Later, the same group has established the inducible cardiac specific expression of the protease 2A. So they found 2A expression in the heart is sufficient to induce dilated cardiomyopathy. So our group also contribute to this area of research by identifying additional substrate within the heart we know play critical role in cardiac function. So we identified at least two proteins. One is called serum response factor, so which is a transcription factor specifically expressed in the heart muscle, and we show this protein is cleaved. This protein play important role in regulation of many cardiac regulatory and contractile genes and some microRNAs. Another protein we identified to be the substrate of the viral protease 3C is called dysferlin. So we collaborated with Dr. Pascal Benages at our center to understand the consequence of this cleavage. And this protein is a muscle-enriched membrane protein. So this protein plays an important role in muscle membrane repair. So a mutation of this protein has been associated with the muscle dystrophy as well as the cardiomyopathy. So our study shows this protein got cleaved after virus infection, leading to the damage of the cardiac function. So another aspect of my research is to understand the role of autophagy in viral myocarditis. So in cardiac disease, much evidence has suggested that proteotoxicity, so which can be defined or measured by the accumulation of protein aggregates as a result of disrupted protein homeostasis, so may represent a common or converging mechanism underlying different types of heart diseases. So using an antibody, A11 antibody, which detects the pre-amyloid oligomers, immunostaining had shown that demonstrates the accumulation of protein aggregates and in both human and animal feeding heart. Also, we know that the damaged protein aggregates often present in the form of ubiquitination. So our own study using the post-mortem human heart from our registry, we found using an antibody detects the ubiquitin conjugates. We show the accumulation of ubiquitin conjugates in diseased heart, including the dilated cardiomyopathy heart or the ischemic heart as compared to a normal heart. So similarly, using the viral myocarditis model, we show the similar observation. Virus infection induces the accumulation of ubiquitin conjugates, and we can also detect the protein aggregates by immunofluorescence staining and using the pre-amyloid oligomer antibody, as well as the disruption or damaged mitochondria in virus-infected heart. So to understand the role of autophagy in viral myocarditis, we first utilize the autophagy GFP reporter mice. So using these mice, we show that after virus infection, the exogenous and endogenous GFP ALC3 lipidation increased, which is a marker for autophagon formation. We also detect the increased GFP positive autophagon puncta in virus infected heart, and also TEM study shows the accumulation of autophagon like double membrane vesicles in virus infected cells. So to understand the mechanism and also consequence of virus-induced the accumulation of autophagons in mouse heart, so we conducted a series of experiments to look at the impact of virus infection on all major steps of all major steps of autophagy process, including autophagon formation, autophagon fused with lysosome, and also lysosome function. Finally, we also look at the selective autophagy process mediated by autophagy receptors. So for this talk, I'm mostly focused on the next part. So I will briefly summarize our major finding. I'm not going to go through all this data we generated for the past many years. So I just quickly go through the major finding and the conclusion we have based on this part of research. So what we found is, number one, virus infection induces autophagosome formation through the non-canonic pathway independent of the ULK and PI3 kinase complex, but requires the PI4K3 beta. So secondly, we found viral infection blocks autophagosome lysosome fusion by cleaving the snail protein, which is important for this fusion process. This snail protein is called Sept29. So then third, we found the virus infection disrupts the lysosome function and also disrupt the autophagic function by cleaving the transcription factor TFAB, which is a master transcription factor, regulates both the lysosome function and autophagy. So, and finally, we found virus infection impair the selective autophagy process by cleaving the autophagic receptor, both P62 and NDP52. So consequently, so increased formation of autophagosome and decreased fusion with lysosome leads to the accumulation of autophagosome within the cardiomyocytes, which benefit for viral replication by providing the replication scaffold. So furthermore, as a consequence of the decreased selective autophagy, the viruses escape autophagy-mediated viral clearance, and also the protein aggregates damaged mitochondria accumulate in the heart, which eventually or ultimately leads to dilated cardiomyopathy. So given the importance of the viral protease, so we collaborate with Dr. Eric Jian and Chris Overall at UBC Biochemistry to systematically identify the viral protease substrate using the technique called TAILS, which was developed by Dr. Overall. So we identified additional substrate of the viral protease, which are involved in many different cellular functions. So move on to the final and also the major part of my talk is on the immune-mediated myocardial damage. So the first evidence of the immune-mediated damage was provided by Doctors Woodruff in 1974. What was found was a depletion of T cells, decrease inflammatory infiltration, and reduce the mortality in this viral myocarditis mouse model. So after this finding, subsequently, there a large body of research have been conducted and provide evidence that both innate and adaptive immune response contribute to the myocardial injury. So despite they recognized the role of the abnormal immune response in viral myocarditis, unfortunately, clinical trials using broad spectrum immunosuppressive drugs failed to provide patient benefits. So back in 1995, there was a famous clinical trial using general immunosuppressive drug. So unfortunately, the data is negative. They show there's no effects on cardiac function, no effects on the overall mortality rate. So even after 30 years, these trials data still valid, still true. So the possible explanation is the general immunosuppression target both protective and detrimental function of immune response. So therefore, it's crucial to identify the specific immune pathway responsible for the adverse inflammatory response. So our recent research focused on the innate immune pathway, which serves as the first line of defense. So we know, unlike the adaptive immune response, many T cells, B cells, and NK cells are involved. For innate immunity, immune pathway, pretty much all cell types in our body are considered as innate cells, including the professional macrophage dendritic cells and non-professional in terms of in the heart, cardiomyocytes, endothelial cells, fibroblasts are all considered immune cells. So in response, so these cells responds to infection quickly and produce cytokine to kill the viruses or facilitate the adaptive immune response. So we know the innate immune cells utilize the pattern recognition receptor to recognize the PAMs, also known as a pattern-associated molecule or passenger-associated molecule pattern, or the danger signals of some host factors, also known as DAMs. So there are three primary pattern recognition receptors: toll-like receptor, Regai-like receptors, and LOT-like receptors. So among three of them, so toll-like receptor and Regai-like receptor are considered as a major pathway, major antiviral host defense pathway by promoting the type 1 interferon production. So in lines of the antiviral role of type 1 interferon, so mice deficient in different sensor or adapter proteins involved in this toll-like receptor or Regai-like receptor pathway, including the toll-like receptor 3 adapter protein or MDA5, IFNA receptor, or the type 1 interferon. So the mice tend to develop more severe myocarditis, suggest a protective function for this sensor or adapter protein in disease progression. So the next question we asked at that time is whether this LOT-like receptor inflammasome pathway play a role in viral myocarditis. So the NLRP inflammasome is a protein complex. So in this complex, there are three major proteins. One is the sensor called NLRP. Is adapter protein ASC, and also the third protein is the effector protein procaspase-1. So in humans, there are at least 22 different members of NLRP inflammasome. So the difference between different members is the structural difference, also tissue distribution difference. So among them, NLRP3 is the mostly extensively studied. So so upon detection of the PAMs or DAMs by the leucine-rich region of the NLRP3, so NLRP3 oligomerized and interact with the downstream procaspase-1 through the adapter protein to form inflammasome. So, abnormal activation of this pathway has been linked to many human inflammatory autoimmune diseases. So, clinical and animal studies have suggested this pathway is indeed activated in the heart of myocarditis. the human study using the analysis of biopsy or autopsy heart from patient with myocarditis shows the increased detection of the ASC aggregates, the red dots here, so which is a marker for the autophagon formation. So animal study also shows the virus infection leads to the increased production and release of interleukin-1 beta in the serum in the heart, as well as in the cardiac inflammatory, infiltrating macrophage. So in mouse model, it was show inhibition of caspase-1 using the peptide inhibitor or the or application of the interleukin-1 beta neutralized antibody. Prolong mouse survival and decrease the cardiac damage and improve the cardiac function. So altogether, this data suggests the role of this pathway in disease progression, and targeting NLRP3 may represent a viable therapeutic approach. So interesting recent study conducted by a group in Europe. So they use colchicine. Colchicine has been widely used for cardiovascular disease as an antiviral drug, anti-inflammatory drug. So they show using this myocarditis model, they show the application of colchicine improves cardiac function and prevent disease progression. So they show at least partially through inhibition of the NLRP3 pathway. So they show application of this drug improves the cardiac function and decrease the release of the cardiac troponin I. So, however, our study using the NLRP3 knockout mice revealed more complex function for this pathway in myocarditis. What we show here is gene-like deletion of NLRP3 actually increase susceptibility to viral infection and cause more severe myocarditis. Here shows increased damage of the cardiomyocytes and also increased infiltration and increased virus load and decreased cardiac function. So I summarize here is we believe during the early phase of infection, NLRP3 play a protective role in host antiviral immunity. So absence of NLRP3 confers host susceptibility. So however, on the other hand, if it's prolonged or persistent activation of this pathway can lead to overactivation of immune response and contribute to disease progression and heart failure. So the take-home message is that while the NLRP3 inflammasome has the potential to be a target, a therapeutic target, caution should be given, especially for people with impaired immune system. So as I mentioned, there are many family members of the inflammasome. So next we want to know what's the role of other inflammasome in myocarditis. So we recently collaborate with Dr. Lila Ho at the Duke National University of Singapore Medical Center to understand the role of other family member of inflammasome. So the first thing we did is to conduct the RNA sequencing analysis to determine which inflammasome is the major inflammasome expressed in cardiovascular system. So we use the primary human aortic endothelial cells or the human iPSC-derived cardiomyocytes. So to our surprising, we found that NLRP3, that's the most frequently studied inflammasome, is minimally expressed in both cell types. Whereas CAR8 indeed is the major inflammasome expressed in the in both endothelial cells and cardiomyocytes. These cells also express the genes encoding other major components of the inflammation pathway, including the gasdermin D, the caspase 1, or the AASC. So Western blot confirms the expression of CAR8 at protein level in both cardiomyocytes, as well as the aortic endothelial cells. But this CAR8 is not expressed in A549 cells, which is a cell line of human airway epithelial cells. So single-cell RNA sequencing analysis of human aortic atherosclerotic plaque tissues also verify that NLRP3 is mainly expressed in macrophages, whereas CAR8 is expressed in both, almost equally important in both endothelial cells and macrophages. So structurally, the CAR8 is quite different from other members of the NLRP3 family. So as you can see here, the CAR8 contains two major domains. One is fine domain, another is CAR domain. So the activation mechanism of CAR8 also is unique, is different from other members. So at basal level, so this CAR8 undergoes autocleavage within the fine domain, so which generate two cleavage fragments. These two cleavage fragments are non-covalently associated with each other. So, but upon stimulation, for example, the CAR8 activator, so that can induce the degradation of the N-terminal fragments. So the N-terminal fragments play an inhibitory role, which binds to the C-terminal fragments to inhibit the function of C-terminals. Once it's cleaved, so the C-terminal fragments is released and binds to the procaspase 1 directly through the CAR domain to form the inflammasome. So mutation of this gene has, the CAR8 gene has also been linked to several autoimmune diseases. So we found virus infection can activate this CAR8 inflammasome as measured by the cell death we observed as the production release of interleukin 18. Interleukin 18, that's a downstream of this inflammation pathway, and also cleavage of gasdermin. Gasdermin is a pore-forming protein. So caspase 1 can cleave gasdermin, leads to the inflammatory cell death. So, and also we found knockout of CAR8 decrease virus-induced cell death and decrease the interleukin 18 production and decrease the cleavage of gasdermin D. So we further studied the mechanism of the virus-induced activation of the CAR8 inflammasome pathway. So first thing we want to ask whether we know viral protease play many different roles. So first question we want to ask whether viral protease is the contributing factor to activate this pathway. So we overexpress 2A and 3C in cells. Then we found that's enough, that's sufficient to induce the production and release of the interleukin 18 to the medium. And a further study, demonstrated that 2A and 3C can directly cleave CAR8, and both can directly cleave the CAR8 and lead to the degradation of the N-terminal fragments through the proteasome and release of the C-terminal cleavage fragments to form inflammasome. So finally, we want to know the function of CAR8 in terms of virus-induced inflammation. So we knock out CAR8 in both aortic endothelial cells and cardiomyocytes, we found that's sufficient to attenuate virus-induced upregulation of many pro-inflammatory cytokines. So here, in summary, based on our data and other people's other findings, we summarize here is internal virus infection differentially activates the NLRP3 and CAR8 inflammasome in different cell types, which collectively contribute to cardiac inflammation. So we know virus can infect both endothelial cells and cardiomyocytes, and leads to the active production, active viral replication, and the productive of the viral protein 2A and 3C, which cleaves the CAR8 and leads to the formation of the CAR8 inflammasome, activation of caspase 1, and production of interleukin-1 beta and 18, and the cleavage of the gasdermin D leads to cardiac inflammation and pyroptosis. On the other hand, the viral particle can be detected by the NLRP3, which is highly expressed in the macrophage, and leads to the activation of the downstream of NLRP3 mediated inflammation. So our data appears to suggest that both CAR8 and NLRP3 can serve as a target for drug development. And I like to point out here that CAR8, all the study we conducted on CAR8 is in vitro using human primary cardiovascular cells, because CAR8 is not expressed in rodent model. So currently we don't have evidence to show CAR8 play a role in vivo because of the lack of the animal models. Another thing I like to point out here is there was a clinical trial using anakinra. Anakinra is an inhibitor for interleukin-1 alpha and beta. So the last month, the clinical trial results just released as at a conference last month. So they show the drug anakinra is safe, but no benefit in terms of reduce the complication associated with viral myocarditis. So anyway, so the bottom line is further study is required to further understand, in addition to interleukin-1 beta, whether interleukin-18 or gasdermin or inflammatory cell death is required, is a target is required to be targeted for the treatment of viral myocarditis. So for the last few minutes, I'd like to give some introduction about our last work, our recent ongoing work on the cGAS DNA sensory innate immune pathway in viral myocarditis. So the cGAS was initially identified as a pattern recognition receptor for during viral infection. They can detect viral DNA. So it was identified in 2013. Later, it was found cGAS can also function as a sensor for tissue damage by detecting dams, including for example, the mislocalized self-DNA, the mitochondrial DNA upon viral mitochondrial damage. So how this pathway is activated? So the cGAS DNA is a sensor which can detect the pathogenic DNA or viral DNA. They can also detect the mislocalized self-DNA, for example, the mitochondrial DNA. So upon binding, which leads to the production of the second messenger cGAM. cGAM can bind to the downstream adapter protein STING, leads to the translocation from the ER to Golgi. So STING can recruit the downstream kinase TBK1 and IKK, and leads to the phosphorylation translocation of the transcription factor, leads to the production of type 1 interference and pro-inflammatory cytokines. So this pathway, this SIGAS-STING pathway has also been linked to many inflammatory diseases, including heart, lung, neural inflammation. So to understand the role of this pathway, we utilize the SIGAS-STING knockout mice. So what we show here is deletion of SIGAS-STING can attenuate the cardiac, both cardiac and pancreatic damage caused by viral infection, and also decrease inflammatory cytokine production. So our study suggests this pathway play a role in disease progression. So in addition to the knockout mice, we also utilize the SIGAS-STING inhibitors. So we got the similar results. So as I mentioned, so in heart, there are many different cell types. So the next question we ask is, what's the relative contribution of different cell types in terms of the cardiac inflammation in viral myocarditis? So first thing we did is to look at the protein level of SIGAS-STING in mouse cardiomyocytes and mouse macrophage. So as expected, we found both SIGAS and STING are more abundantly expressed in macrophage as compared to cardiomyocytes. So more interestingly for us is we found virus infection can induce the upregulation of pro-inflammatory cytokines only when cardiomyocytes and macrophage are co-cultured, so but not individually cultured. So we know that virus can only effectively infect the cardiomyocytes, but not macrophage, as measured by the production of viral protein VP1. So together, our study suggested that there was a crosstalk between cardiomyocytes and macrophage. So the crosstalk is required for the cardiac for cardiac inflammation. So we also conducted some experiment to study the crosstalk between cardiomyocytes and macrophage in terms of the activation of the SIGAS-STING pathway. So we know internal virus is like SARS-CoV-2, they both are RNA viruses. So we do not expect RNA virus can directly activate the SIGAS, because SIGAS is considered as a DNA sensor. So, but we know virus infection can induce the mitochondrial damage. So we want to know if virus infection can lead to the release of mitochondrial DNA from mitochondria to cytosol or in the supernatant. So we conducted immunostaining using the double-stranded RNA antibody and also TOM20 antibody. TOM20 is a marker for mitochondria. We show the release of double-stranded DNA from the mitochondria. We also conducted the RT-PCR to measure the mitochondrial DNA expression in the supernatant of cultured cells. We show virus infection increase the release of mitochondrial DNA. So furthermore, we show that treatment of macrophage using STING agonist, diABZ or CGAM, is sufficiently to induce the phosphorylation of STING and its downstream TBK1 in a dose-dependent and time-dependent manner. So finally, we treated the macrophage with SIGAS agonist hearing tested DNA. So it shows it sufficiently significantly promote the production of SIGAS-STING related pro-inflammatory cytokines. So our data summarized in this diagram here. So virus infects the cardiomyocytes, can lead to the mitochondrial DNA release, and treatment of the macrophage with either C gas agonist or the STING agonist can activate this pro-inflammatory gene expression. So this is my last slide. This is still ongoing project, but based on what we know so far, this is the model we proposed. We think virus can effectively infect the cardiomyocytes, leads to the damage of mitochondria and release of mitochondrial DNA into the cytosol, and that activates the gas, leads to the production of CGAM. And macrophage can take up the, once the cardiomyocytes die, or mitochondria can take up the mitochondrial DNA or the CGAM through the phagocytosis and leads to the activation, triggers the CGAS-STING pathway, leads to a chronic inflammatory response, eventually leads to the cardiomyopathy phenotypes. Based on our data, we expect genetic deletion or pharmacological inhibition of this pathway can be a treatment, can attenuate the cardiac inflammation and improve cardiac function. So finally, I'd like to acknowledge the people involved in this project. So Junye and Yasa, he both work on the autophagy project, and Dr. Chen, she work on the NRP project, and Yasa and two co-op student and team, they contribute to the paper on the CAR8 and the project on CGAS-STING pathway. And also I'd like to thank our collaborators and also the funding agency, the funding from Sept and Harvard Stroke Foundation. Thank you for your attention.
Thank you so much. So we have a little bit of time for questions here. I'm going to start off. I mean, there's obviously a huge amount of excitement with STING antagonists, and also for other clinical uses, STING agonists are being developed. But how far would you be aware? What levels of evidence would you need to acquire for a clinical trial? Because it seems like past clinical trials haven't been that effective. Other approaches to ameliorate, to dampen the immune response to infection, but this is a very logical approach. So where are we in terms of
actually this you mean this CGAS-STING project? Actually, this project was just funded by Sept, so people reviewers got so excited based on our preliminary data. So what we propose from here, what we propose to do is to look at the role of the macrophage and cardiomyocytes in vivo. So what we want to do is generate the cardiac myocytes specific knockout of CGAS and macrophage specific knockout of CGAS-STING to look at what's the role in animal models. In animal models first. And another challenge part I don't have time to share is we found this pathway actually, if it's overactivated, it's a balance. They have antiviral role, it's not surprised. So at the early phase, this if we activate this pathway, actually for SARS-CoV-2 also show that if early enough we activate this pathway, actually we can attenuate viral replication. And later stage, if it's sustained or prolonged activation is bad to our body. So it's a long way to in terms of clinical trial, we need to have a more animal study to move on to the clinical trial. But the challenge part, another challenge part, as I mentioned earlier, is mouse and human, they have different inflammation pathway in terms of NLRP3. Bat, they have very low. I think bat, the system bat, they have very low NLRP3. And some animals, they do not have the CAR8, the rodent model. So that's why that explains maybe the animal model, some animal model, they do not develop very severe viral myocarditis. So it's still quite challenging for us. Right now, we work with Dr. Yin Wang, so to use the human registry samples to hope to provide some human evidence to show this pathway is important in terms of viral myocarditis.
Thank you. And it's great to see that you're using your obvious expertise in viral infection, work on oncolytic virus as well, which we'll have to hear. Speak up. I can kind of monitor for questions, but if anybody has any questions, please ask. On where people can contact you directly. So at this point, I'd like to thank you, Honglin's wonderful research and the journey which you've taken and the direction you're going, which I can see why CIHR are so excited. And next week is going to be a panel session, and Sneha will send an alert around the topic and the speaker early next week, and look forward to seeing you all at future rounds. So thanks so much, and enjoy the rest of your week.
Okay, thank you.